Hi, I'm Guido, and if you don't know me, I did a postdoc with Zach at Champalimaud, and today I'll tell you about my work there, which is on the best neuromodulator, serotonin. I'm always a bit jealous when I see these dopamine talks that are all so nice and clean. Serotonin is a bit more chaotic, if you will. So previous work in the lab has shown that serotonin neurons are driven by surprise or unsigned prediction errors. So if you couple an odor, in this case, with an outcome, that could be a reward or an air puff or nothing, and then you reverse the contingencies and you measure dopamine with vibrophotometry in the dorsal-raphe nucleus, you see that after a reversal, there's an increase in serotonergic activity, and this is basically both the case for positive and for negative prediction errors. So basically when an odor that was previously associated with an air puff is suddenly associated with a reward, you see this increase in serotonin. But also when an odor that was previously associated with a reward is suddenly associated with nothing, you also see an increase in serotonin. So it's this case where like both positive and negative outcomes or more positive or more negative outcomes result in an increased serotonin signal. So we thought that, this means that if you stimulate serotonin in the IBL task, we see, yeah?
So not negative stimuli, just no stimuli.
Exactly, yeah, no outcome. So because of this, we thought if we would stimulate serotonin in the IBL task, we could maybe see that you make trials artificially surprising. So this would result in an increase in flexibility, for example, as indicated by faster changes of the behavior after a block switch, or a change in how the animals are incorporating prior knowledge with current sensory evidence. So I used channelrhodopsin, injected it in the dorsal-raphe nucleus, and implanted a fiber over it to be able to stimulate serotonergic neurons in a SERT-CRE mouse. And then I stimulated the serotonin in this way. So basically the optogenetic stimulation started with the onset of the visual cue, and it continued up until the moment that the animal received the reward. And this happened in blocks of trials. So there's the bias blocks, of course, if you can see here on the right bottom. And then there's also these blocks of serotonergically stimulated trials, and the two are uncoupled so that the animal can't really infer anything about the block structure from which trials are stimulated and which are not. Okay, so first thing we looked at was the psychometric performance. So here I plotted, for one example mouse, the psychometric curves for the right blocks in purple and the left blocks in orange. And the dark line is the serotonin-stimulated trials, and the light lines are the non-stimulated trials. And as you can already see, not much is going on. So you can pull out a couple of metrics from this, like the slope and the bias and the lapse rate. Before you ask, Alex, no, I did not quantify the bias in two different ways, like horizontal and vertically. I learned about this yesterday, but I'll do that. Anyway, there was no effect. So if you then look over animals, here every dot is an animal. There's no change in performance, no change in the slope. Well, there was a trend for the slope, but it wasn't significant. No change in the bias, and no change in the lapse rate. So that was quite disappointing. So of course, we did some more digging. So then we looked at, reaction times, also no effect. So then we looked at what happens at the block switches to see if they're maybe more flexible. So the easiest way, I've done this in various ways, but the easiest way to plot this is just to plot the fraction of choices towards the side that will have the high prior probability. And then you see there's no difference. So basically they change their behavior after a block switch equally fast in the 5-HT stimulated trials and the non-5-HT stimulated trials. So then we thought maybe the effect is more nuanced. Maybe it only happens at certain contrasts, or maybe it has something to do with reinforcing previous choices. So I fitted the probabilistic choice model from the behavior paper with a bunch of weights. So there's the visual evidence predictors. So there's a separate predictor for each of the stimulus contrasts. Then there's the past choice, whether it was left or right. And then there's also a predictor for the prior the block prior basically, and just a constant. And these were separated out by 5-HT stimulated and non-stimulated trials, so that there's a weight for both, for the trials that were stimulated and the trials that were not stimulated. And this goes into the probabilistic choice model, and it outputs its prediction basically. So yeah, when we look at the weights, we see what we expect, and we also saw this in the behavior paper. So there was a nice replication of this. But then when you look at the 5-HT stimulated trials, again, there was no difference. So yeah, at this point, I started to get pretty disappointed that there didn't seem to be any effect of the serotonin stimulation on the behavior. So then, we were, thinking what to do. And then we thought about this other piece of data from the lab. That's with mice that are running around in an open field. And when you stimulate serotonin, when they're just running around this box, they slow down. But when you stimulate when they're actually shuttling back and forth to reward points, and you stimulate then, they don't slow down. So if they're engaged in a goal-directed action, the serotonin stimulation has no effect. But if they're just sort of chilling and just walking around, then it does. So the effects are very context dependent. So then we're like, okay, this is interesting. Maybe we should focus a bit more on the things that happen outside the task. So after the task, there's a period where the animal is not doing anything. It's the spontaneous activity period. And also there, I've stimulated serotonin. So when you look in this part of the recording, so after the task, and now the animal is just sitting, not doing anything, you can pull out several features from their behavior from the camera, like the pupil size and the whisking and the sniffing, and align them to the times of the optogenetic stimulation. And you do see quite strong effects here. So Fanny has already shown that serotonin stimulation induces increased pupil size, which we replicated, but also it induced whisking and sniffing, which are more like, I guess, exploratory behaviors. Yes? Yeah, well, it depends on how you think about that, but because it could also be that the animal is basically slowing down to explore the open field more, to explore what's around it, basically. So it depends on the interpretation, I guess.
They whisk, sniff more, and get bigger pupils in the open field.
We haven't checked. It's quite hard to do that with freely moving animals. But as far as I know, we didn't check that. And also one other interesting observation is that there was a reduction in hippocampal sharp wave ripples. So you might know that in more offline states, there's these sharp wave ripples that are associated with memory consolidation. So when there's fewer sharp wave ripples, it means that the animal is more sort of maybe engaged in the present moment, and it's sort of not in this offline dreaming state, if you will. We then did neuropixel recordings to see what happens in the brain at this time. I did 86 neuropixel recordings in 17 mice, and in total recorded 7,500 good neurons. And in the brain, we also saw, quite some changes in neuroactivity. So there were some neurons that would increase their activity when serotonin was stimulated. There were other neurons that were decreased. And in total, there were quite some neurons that were modulated in this way, between 20 and 40%, depending on the region. And the regions that were most strongly modulated were hippocampus and medial prefrontal cortex. And 20 to 40%, I think it's quite high overall. The sign of the modulation, so you can already see they go both up and down. If you take the average modulation index over all the neurons in a region, you see that some regions on average are excited, like the periaqueductal gray, medial prefrontal cortex, orbital frontal cortex. Some other regions are suppressed, like the hippocampus, for example. But there's also all these regions in the middle. And if you now plot this, but then per neuron, so here now every gray dot is a neuron on the right, you see that those regions in the middle, they actually do have quite some modulated neurons, but they're bidirectional. So half go up, half go down. So the population mean is somewhere in the middle. But it doesn't mean that nothing is happening in these regions. And also the ones that are excited, like the PAG, you still see quite some neurons that are actually suppressed. So there's a lot of variability here from neuron to neuron. Okay, so at this point you're probably confused. So yeah, we were also confused. So yeah, how is it possible that we see these like big changes in neuroactivity when you stimulate serotonin? We see these big changes in their sort of like innate behaviors during the quiescent period, but there was no effect at all on the behavior during the task. And we were scratching our heads.
So, Guido,
Yeah.
did you look at neural activity during decision making?
Yes, yeah, I didn't have time to put a slide in, but it's basically the same. Yeah, so it's very similar to during a quiescent period. Yeah, but also during decision making, you see these strong changes in the. Yeah, it was basically it looked very much like this. Yeah, very much like this, slightly less strong, but yeah. So also during the task, we saw these changes in neuroactivity, but no change in the behavior. Okay, so then we sort of turned to the manifold analysis from the brain-wide map to see maybe if that could shed some light on why this is happening. So here I constructed these really large arrays with like the trial average activity of each neuron pooled across neurons, across sessions, across mice into one very large multidimensional array. So this is across all, it's basically pooling all the neurons from all the animals together. And you can split the trials in several different ways in my case. You have the left and right choices, but you also have the serotonin stimulation or no stimulation. And the first thing I did was just replicating what has been done in the brain-wide map paper, and that's splitting by choice. So the neuroactivity is aligned to the first movement of the wheel, which is 0 seconds. And then I go back in time 300 milliseconds before the choice is made, basically. And you can see these nice choice trajectories that we already saw also in the brain-wide map paper. And you can quantify the choice separation by taking the Euclidean distance between each point in neural space. And then you see something very similar that we saw in the brain-wide map paper as well. This choice separation increases when it comes closer to the decision point. And if you split it out per region, if you do it per region, you basically see the regions that you'd expect, like M2 and MPFC, and also a bunch of other ones. Okay, so then you can also split by stimulation, right? So now I just pooled all the trials together. They were stimulated, regardless of choice, and non-stimulated. And you see something similar, but less strong. So there's also these two trajectories, and there's this separation, and it's significant, but it's like not as strong as the choice separation. And it was mostly in areas like MPFC and hippocampus. So I must say that the gray shaded area here is the shuffle control. So the choices are shuffled like in the brain-wide map paper per block. And in the case of the 5-HT stimulation, I used the pseudo-session approach. Okay, so then what happens when you split out for both choice and serotonin? So here now we have 4 trajectories for each of the cases. And they also similarly diverge over time for the left and right choices. But there's now also this second axis of the stimulation, basically. And I noticed that the choices, they seem to separate out over one axis, and the serotonin seems to separate out over another one. So to quantify this, I took basically the last point before the choice, and then took these trajectories, the points of these trajectories, and got like an average choice axis, which separates the right choices from the left choices, and the serotonin axis, which separates the serotonin ones, the top ones, from the bottom ones. And then you can quantify the angle between these two axes, basically. So the question is like, okay, are these more orthogonal than you expect by chance? So I calculated the dot product. So this is 1 minus the normalized dot product, and I call this orthogonality. And then I also do this for the shuffled control, and we see indeed that the sort of like the geometry between the choice and the serotonin axis is more orthogonal than you would expect by chance. And it's also like leading up to the choice, it's increasing, basically. And we saw this geometry mostly in medial prefrontal cortex, visual cortex, and secondary motor area. Right, so how we think about this is that it's sort of like giving a way for the brain to have an extra sort of like dimension of information that can be read out by downstream target regions in several different ways, basically. You have these 4 sort of like trajectories, and you can basically project them onto the yellow plane here, and then you can see the choice separation, so left versus right. But you can also project them over this other dimension, this other axis, and then you basically get the serotonin trials and the non-serotonin trials. But because they're orthogonal, they don't actually interfere with one another. So that means that serotonin can signal things like whatever it is it signals, maybe state or motivation or whatever it is, without actually interfering with the goal-directed behavior. So it's giving the brain this sort of like extra way of putting some extra information on top of the choice to signal other things that it wants to signal. I'm done. So to conclude, serotonin stimulation induces an online behavioral state during quiet wakefulness, and it modulates brain-wide neuroactivity in a bidirectional way. This behavioral state doesn't interfere with the decision-making, and that's probably because it's modulating these neural dynamics in an orthogonal subspace relative to the choice factor. Okay, with that, I want to thank Zach and the other people who did the work that are in bold here, and a special thanks also to all the staff, and thank you for listening.